United States, and then a sabbatical on the Albert Einstein College of Medicine. Presently, she's head of the Laboratory of Epilepsy and Therapeutic Strategies at the Department of Acute Brain and Cardiovascular Injury at the Mario Negri Institute for Pharmacological Research in Milano, in Italy. Her main research focus is on cellular and molecular mechanisms of seizures and epileptogenesis, and also the development of new markers and new biomarkers and treatment. She authored more than 200 papers. I should say, I checked yesterday, it's 282 papers. She has an age of 90 and several book chapters, reviews, etc. Is a past chair of the Neurobiology Commission of the ILAE, of the International League Against Epilepsy, and she belongs to several commissions of the ILAE, received several prizes, etc. So thank you, Annamaria, and it's a real great pleasure to have you here for us today.
Thank you very much, Alberto, Luisa, for this very kind invitation. I'm very happy to give this seminar, and I hope it will be clear, and I will be ready to take questions at the end of my presentation. So I hope that you see my slides well. Is that fine? I mean, can you see the slides?
Yeah,
Yes.
perfect. Yeah.
And also the pointer, right? The arrow.
Yeah, perfect.
Okay, good. So I will start with this presentation. Okay, by showing you the updated classification of the epilepsies according to the International League Against Epilepsy. And I would like to highlight that this updated classification takes into careful account the etiology of the various forms of epilepsy. And looking at the etiology, you can see that the inflammatory brain response is associated with many of them. It was historically first identified in Rasmussen encephalitis, but then with the increased knowledge in the field due to the ability of assessing brain specimen resected as surgery from various drug-resistant forms of epilepsies, it was possible to broaden the presence of this brain inflammatory response to many other etiologies, which include the structural epilepsies, that are mostly drug-resistant forms of epilepsies, and also some genetic forms, and epilepsy syndromes such as the developmental and epileptic encephalopathies, which include the Dravet syndrome. I will talk more specifically about the neuroinflammatory response in epilepsy, which is a tissue response, a brain tissue response due to the activation of innate immunity mechanism. So as you know, innate immunity generates rapid and non-specific inflammatory responses to signal that are presented to innate immunity receptors expressed by immunity cells, and these are called pattern recognition receptors. These receptors are very interesting because they can recognize pathogen-associated molecular patterns that are molecular structures expressed by pathogens. But interestingly, they can also recognize the so-called damage-associated molecular pattern that are molecular motifs that are expressed by molecules released because of cell death or cell activation, tissue injury, and also during recurrent seizures. So in so-called sterile conditions, in the absence of an identified pathogen. So as you know, this innate immune response is the first line of defense against invading pathogens, and also to reestablish homeostasis after tissue damage or injury. And it's very important to initiate also specific adaptive immune responses. When we talk about neuroinflammation as a consequence of innate immunity activation, we don't have to think only about two blood innate immunity cells, but mainly to brain parenchyma cells, which include astrocytes, microglia, neurons, cell component of the blood-brain barrier, because these cells are pivotal for the activation of the so-called cytokine storm that is upstream the neuroinflammatory cascade due to the transcriptional activation of inflammatory genes. So this schematic drawing shows you the neuroinflammatory response, which is, can be considered now as a common brain response in drug-resistant forms of epilepsy. So this response consists of the induction and release of a variety of inflammatory molecules by brain parenchyma cells, and is strictly associated with evidence of oxidative stress in the tissue. These molecules are upregulated together with their receptors and cell signaling, and some of the molecules can also be collected in the bloodstream, therefore representing potential biomarker of this neuroinflammatory response. There are several triggers of the neuroinflammatory response as listed here. Can you hear me well? Yes, okay. So there are several triggers of the neuroinflammatory response that also represent risk factors for the induction of epilepsy. And among those, I would like to stress, as I said before, the release from the brain tissue of disease proteins, so-called danger signals, together with inflammatory cytokines. Two prototypical signals of this kind are the pro-inflammatory cytokine interleukin-1 beta and the danger signal high mobility group box-1 that are inflammatory molecules that have a pivotal role in the induction and maintenance of seizure activity as shown by pharmacological interventions in animal models. So in the presence of release of danger signals, we can talk about a sterile form of inflammation that is a result of different types of injuries in the absence of demonstrated pathogen. Importantly for epilepsy, there is also the concept, the demonstration that the neuroinflammatory response in brain tissue can also be evoked by a state of enhanced neuronal activity, such as occurring during seizures. And indeed, recurrent seizures, even in the absence of neuronal cell death, can induce a very rapid and long-lasting neuroinflammatory response. Just a few examples of what I'm talking about. And also, I would like to stress the concept, the new concept, that the neuroinflammatory response in human brain tissue, as shown here, is strictly associated with evidence of mTOR activation in structural epilepsies. So the so-called mTORopathies, which includes malformations of cortical development, have been demonstrated in the epileptogenic brain areas to express markers of both mTOR activation and neuroinflammation. So here we have an example, immunohistochemical evidence of induction of interleukin-1 beta in brain-resected epileptic foci from mesial temporal lobe epilepsy patients. So these are evidence from human tissue. And you can see in brown the cytokine that is induced in perivascular, astrocytic end feet, and in parenchymal glia, including reactive astrocytes and microglia. In malformations, cortical development, particularly focal cortical dysplasia type 2B, that is characterized also by the presence of balloon cells, we can see upregulation of these inflammatory cytokines in the balloon cells that are highly immunoreactive cells and are highly epileptogenic, as well as in the dysmorphic neurons and in parenchymal glia. The same cell types that express the inflammatory molecules also express the relevant receptors, depicted here by IL-1 receptor type 1 that mediates all the biological action of interleukin-1 beta. And the same occurs for the cell signaling, therefore denoting that upon their release, these inflammatory molecules can mediate both autocrine and paracrine effects. As I mentioned before, there is also evidence for mTOR activation, and I would like to bring to your attention this interesting example. This is a tuberosclerosis complex fetal brain lesion at the 23rd week of gestation, where in the deep white matter, the lesion is associated with strong evidence of activation of innate immunity mechanism, receptors such as the pattern recognition receptors and inflammatory molecules. I anticipated that these inflammatory molecules in animal models have been shown to contribute to seizure events. Therefore, this is a nice example of a neuroinflammatory response in a fetal brain that is going to develop, in the majority of cases, drug-resistant forms of epilepsy, highlighting that the neuroinflammatory response can precede the onset and recurrence of drug-resistant seizures, as it has also been shown in animal models. So now there is quite solid evidence for an interaction between mTOR activation and neuroinflammation that can converge onto a common epileptogenesis mechanism that are listed here and that we'll touch briefly upon during the next slides. In animal models, the mTOR activation can be induced in conditional knockout mice where the amartin gene can be deleted predominantly in GFP positive astrocytes. So if you induce deletion in astrocytes of this gene that negatively regulate mTOR activity, you have an upregulation of mTOR and you induce a model of mTORopathy in animals. In these animals, you see that there is an inflammatory response in astrocytes that can be observed in vivo, but also in vitro by culturing astrocytes taken from the mouse brains. And immunohistochemistry has shown that this inflammatory response is mostly induced in reactive astrocytes. And these reactive astrocytes with an inflammatory phenotype show evidence of this regulation because in these knockout animals there is a strong reduction in the expression of the glutamate transporter. As you know, the glutamate transporter in astrocytes is pivotal for the removal of extracellular glutamate. Therefore, a reduction in the expression of this important transporter leads to an increase in extracellular glutamate, which is permissive for seizures. So this is something that has been also established in other experimental contexts, that the inflammatory phenotype of astrocytes is linked to evidence of astrocytic dysfunction of the homeostatic properties of astrocytes that are so important for maintaining a physiological level of activity in neuronal networks. Again, stressing the association between neuroinflammation and mTOR hyperactivation, these animals with the activation of mTOR in astrocytes develop seizures after the first 3 postnatal weeks. And these spontaneous seizures can be strongly inhibited both by anti-inflammatory intervention. This is done with a drug that is the broad antagonist of cyclooxygenase, and also using rapamycin, that is, as you know, an inhibitor, specific inhibitor of mTOR. Okay, so to sum up what has been published in the literature since the last 20 years, now we can take this as solid evidence that neuroinflammation is present in epileptic foci from several forms of structural drug-resistant epilepsies, as well as in Rasmussen encephalitis, and also in autoimmune limbic encephalitis. And most recently, there is also evidence for the presence of a neuroinflammatory response and its contribution to seizures in patients with a clinical presentation of new onset refractory status epilepticus and its subtype FIRES. I will go into the detail later on. And the neuroinflammatory response is also mimicked in experimental models of epilepsy, of acute seizures and epilepsy, both in adult and in mature rodents, and again, more recently, also in models of genetic forms of epilepsy, such as absence seizures, Dravet syndrome, and progressive myoclonus epilepsies. So why this neuroinflammatory response persists in the epilepsy brain tissue? I mentioned at the beginning of my presentation that is an homeostatic response to either tissue injury or infection. So it should reestablish the homeostasis of the damaged tissue. However, it has been well established that if this neuroinflammatory response is not well controlled, persists for too long or is too intensive, then can lead to tissue damage and dysfunction. And indeed, there is now evidence, both in human epilepsy tissue and in experimental model, that in epilepsy there are inefficient mechanisms for the resolution of the neuroinflammatory response. So the resolution of the neuroinflammatory response can be induced by the upregulation together with the inflammatory molecules of anti-inflammatory counterparts. And these anti-inflammatory molecules, that some of which are listed here, are induced to significantly lower extent in epilepsy tissue than their pro-inflammatory counterparts. Why should be the opposite? They should be predominant in order to rapidly block the neuroinflammatory response after its onset, to maintain its homeostatic function and prevent the detrimental consequences. Moreover, there is also evidence for both transcriptional and epigenetic mechanisms that are deregulated and can contribute to a persistent neuroinflammatory response by promoting the transcription of inflammatory genes, by acting on the activation of promoter of inflammatory genes by, for example, hypomethylation of such promoter, as shown for IL-1 beta in tuberous sclerosis tubers, and also by deregulation of microRNAs. MicroRNAs, as you know, are small non-coding RNA that regulate protein levels in the cells by modulating post-transcriptionally the level of the transcripts. So this microRNA can be produced by neurons, but also by astrocytes and microglia. And there is evidence in preclinical model, also supported by validation in human epilepsy tissue, that some microRNA are deregulated in epilepsy that are produced by glial cells, by inflammatory glial cells, and this might contribute to the dysregulation of downstream protein that are important for hyperexcitability phenomena. In human epilepsy tissue, these are data from the evidential. There is evidence for differentially expressed microRNA in temporal lobe epilepsy human tissue as compared to control specimens, some of which are pivotally involved in the control of inflammatory signaling that are involved in seizure mechanism. So if this neuroinflammatory response is detrimental and has some role in the onset, recurrence of seizures and the neurological outcomes in epilepsy patients, of course, an important question is, can we treat this neuroinflammatory response and improve the disease outcomes? The preclinical data give a positive answer to this important question, and there is also some initial clinical translation supporting the preclinical studies. However, the inflammatory process is very complex, and we have to take into account that we have to know very well the dynamics of the neuroinflammatory response, because both microglia and astrocytes can present a continuum of phenotype that includes, this is depicted for astrocytes, not only pro-inflammatory mediators, but also anti-inflammatory mediators or immunomodulators. Therefore, the challenge here is to target these cells when they are in their predominant pro-inflammatory state, and to target those inflammatory pathways that are pivotal for the pathological consequences that we want to target. And there is a literature supporting that during the epileptogenesis process, reactive astrogliosis is predominantly detrimental, and preventing the dysfunctional phenotype of astrocytes that are reactive and inflammatory during epileptogenesis can increase the seizure threshold in animal models and reduce the recurrence of seizures. Astrocytes are really pivotal cells for sustaining the neuroinflammatory response in epilepsy, and there is evidence both in human diseases condition and in experimental models that there are several homeostatic functions of astrocytes that are dysregulated in epilepsy, leading to extracellular changes in potassium, water, glutamate, and inhibitory metabolites such as adenosine that promote an extracellular environment that is permissive for seizures. So, animal model evidence. Of course, to understand which is the role, so to say, of neuroinflammation in seizures and epileptogenesis, the animal models have provided pivotal information since one can intervene with pharmacological means or genetic manipulation on specific inflammatory pathways to understand which is their contribution to epileptogenesis and to establish seizures. And by doing so, I summarize the 20 years work in this slide. It is possible to state that, preclinically speaking, neuroinflammation contributes to seizure recurrence since anti-inflammatory interventions can reduce acute symptomatic seizures, can reduce status epilepticus, can reduce chronic established seizures. But neuroinflammation can also be considered a disease modifier in models of epileptogenesis, where an acute brain injury leads through a so-called latency period to modifications in the brain tissue that are generating spontaneous recurring seizures and accompany the progression of the disease. Since anti-inflammatory intervention before the onset of spontaneous seizures in this acute brain injury model, but also early on after disease onset, can modify the disease course. And importantly, there is also preclinical evidence for the involvement of neuroinflammatory processes in the mechanism of drug resistance, particularly for drug refractory status epilepticus. I've listed here most of the mediators that have been targeted pharmacologically or genetically in this context in order to support the statements of this slide. So I try to summarize some of the preclinical trials and also the clinical studies in support of therapeutic potential of specific anti-inflammatory treatments in epilepsy. If you focus on preclinical trials in models of acute brain injury leading to chronic seizures, the interventions with specific anti-inflammatory drugs at various phases of the disease have provided significant therapeutic effects, which do not only involve the reduction of seizures, but also neuroprotection and improvement of comorbid behaviors, which is a very important feature for patients affected by epilepsy, by drug-resistant epilepsy. And also clinical studies, proof of concept, initial clinical studies, targeting some of the inflammatory pathways identified in animal models as involved in seizures, have shown signs of therapeutic effects in patients with drug-resistant epilepsy. I would like to focus a little bit now on repurposing drugs for therapeutic effects in epilepsy. Why so? Because in the context of inflammation, we are relatively lucky. There are many anti-inflammatory drugs that have been developed for other medical conditions, particularly for autoinflammatory and autoimmune disorders. I have highlighted here some of these drugs, both anti-inflammatory and antioxidant. The problem is that in most of these conditions, I mean, the anti-inflammatory intervention was not meant to target the brain, but the peripheral inflammatory environments. And therefore, some of these drugs do not have a good permeability to the brain. We have to take into account that there is an impairment of blood-brain barrier permeability in drug-resistant epilepsy, but some of these drugs are biologicals, are big molecules, and therefore it would be important to find mimicry of these drugs with higher permeability or to modify them in order to make their access to the brain facilitated. Anyhow, I would like to give you two brief examples of repurposed drugs, dimethyl fumarate and fingolimod, that are drugs in medical use for multiple sclerosis, for example. Dimethyl fumarate is very interesting because it has both anti-inflammatory and antioxidant activity. Its antioxidant properties are mediated by its ability to promote the nuclear translocation from the cytoplasm to the nucleus of this transcriptional factor. It is very important for activating detoxifying and antioxidant enzymes. Fingolimod not only inhibits the aggression of T cells from the lymph nodes, but also activates the sphingosine 1 phosphate type 1 receptors that are enriched in astrocytes. And upon their activation, they switch the inflammatory phenotype of astrocytes towards an anti-inflammatory condition. Both drugs have been tested in animal models according to two main protocols, what I call early intervention or late intervention. Early intervention means that the drugs were given to the animals after the acute brain injury that was provoked by status epilepticus induced by cyanuric acid. And the treatment was given to the animals before they developed the spontaneous seizure, so in the prodromal phase of epileptogenesis for a limited period of time. Why a limited period of time? Because the authors wanted to assess the so-called disease modification effect of the treatment. That is an effect that should persist in the absence of the drug itself, after the drug was washed out. So with this early intervention, with dimethyl fumarate, indeed, you can see that as compared to the natural history of the disease in vehicle-treated animals, there was a drastic reduction in the frequency of seizures, in the number of seizures per week, that was maintained for several weeks after drug washout, because the drug was given only for 1 week. And there was also an increased percentage of animals that were seizure-free. If the drug is given when the animals have already developed the chronic spontaneous seizure, so in the chronic phase of the disease, not in the prodromal phase, again, the drug was still effective. So what the authors did was to record the baseline of spontaneous seizures in the chronic epileptic animals, and then they gave the drug for 1 week. And again, there was a drop in the frequency of spontaneous seizures that was maintained over time, even in the absence of the treatment. And similar results were observed using fingolimod, another repurposing drug. Again, early intervention before the animals develop epilepsy was able to reduce more than 50% the frequency of seizures, an effect that is maintained also after drug washout. Late intervention, when the animals have already chronic recurrent seizures, again was able to decrease in a dose-dependent manner the frequency of seizures. So very encouraging results with drugs that have been already tested in humans, and therefore the clinical translation to epilepsy could be facilitated. What about new investigational drugs? We have done an experiment with an intervention at the disease onset. with the intent to test whether the progression of the disease could be blocked or improved if the drug is given promptly after the animals develop the first spontaneous seizures, not in the chronic epilepsy phase. And we did this so with a combination of anti-inflammatory drugs targeting the biosynthesis of IL-1 beta and the toll-like receptor 4 that are pattern recognition receptors activated by another inflammatory and nicotogenic molecules danger signal that is high mobility group box 1. Why we combined? We combined because we wanted to maximize the chances of therapeutic effects since the neuroinflammatory response is complex and several pathways are simultaneously activated. So if you focus on this part of the slide, the bargrams are the seizures occurring in the animals during the disease development, with day 1, the day of seizure onset. So we treated the animals at the onset of epilepsy for only 9 days, and then we withdraw the treatment and we looked at the chronic phase. And as you can see, there was a massive decrease in the frequency of daily seizures when the animals were treated early at the disease onset with anti-inflammatory drugs. An effect that was not observed if you use a classical anti-seizure medication such as carbamazepine. What about treating with new investigational drugs chronic drug-resistant seizures in animals that have already developed the disease? So we use a model of intramygdala kinase injection because in this model the seizures are resistant to various anti-seizure medication, therefore providing a model of drug-resistant epilepsy. And these seizures, in spite of being poorly controlled by several anti-seizure medication, were highly responsive to various anti-inflammatory treatments targeting different pathways. An example is the IL-1 receptor antagonist that we tried in chronic epileptic animals, providing a significant 50% reduction on average of seizures. The treatment was given for 2 weeks and then withdrawn. And it was not enough to provide disease modification effect, but rather acute symptomatic effect on seizures, because the seizures were restored upon drug withdrawal. So it is possible that in the chronic epilepsy phase, the drug should be tested for a longer treatment duration in order to provide a disease modification effect. Hints into disease modifying effects have been given when trying P2X7 receptor antagonist, since in this case the anti-seizure effects in the chronic phase persisted for at least 10 days after drug washout. So neuroinflammation, as I mentioned before, is involved also in drug-resistant mechanism. This has been mostly studied in drug-resistant status epilepticus that can be induced by either chemoconvulsive drugs or electrical stimulation in rodents. Why so? Because status epilepticus is, in these models, resistant to benzodiazepine and other anti-seizure medication. But if the animals are treated with anti-inflammatory drugs targeting these pathways, then the status epilepticus become susceptible to this anti-seizure medication, indicating that this neuroinflammatory response evoked in status epilepticus is involved in the mechanism of drug resistance. And indeed, there is a prominent neuroinflammatory response in status epilepticus model, as shown here by pathway analysis after RNA-seq in the hippocampus of these animals subjected to status epilepticus induced by electrical stimulation. And immunohistochemical analysis again showed that this neuroinflammatory response mainly involved reactive microglia and astrocytes, as well as a cell component of the blood-brain barrier. And this led to a very promising clinical translation of preclinical findings. Why so? Because we know that the neuroinflammation is triggered by drug refractory status epilepticus, and we have tested first, and this was confirmed by colleagues in the literature, that the IL-1 receptor antagonist, anakinra, reduces status epilepticus incidence and severity and improves its responses to anti-seizure medication. Anakinra is an endogenous molecule, is a peptide that is produced during the inflammatory response and is a competitive antagonist of IL-1 receptor type 1, therefore preventing the action of the endogenous IL-1 beta. So this led to the translation of this treatment to patients with new onset refractory status epilepticus. Why? Because in these patients there is evidence of a prominent neuroinflammatory response during status, since there is an intrathecal production of several pro-inflammatory cytokines that have ictogenic activities in the animal models. And also the analysis of autoptic tissue from patients who died in status epilepticus showed the presence of reactive glia and ictogenic cytokines in the epileptogenic circuitry. And indeed, now anakinra is used in the patients with NORSE, and retrospective analysis of therapeutic effect have shown that if anakinra is given within the first 2 weeks from seizure onset, can control seizures in up to 70% of patients that otherwise would have seizures refractory to any other anticonvulsive treatments, including large broad spectrum anti-inflammatory drugs such as steroids. Towards the end of my talk, I would like also to briefly touch upon biomarkers of neuroinflammation for examining the epileptogenic lesions and possibly stratifying the patients that would best benefit from an anti-inflammatory intervention. And there has been progress in neuroimaging by developing ligand, PET ligand, that can image the epileptic focus that corresponds with this imaging analysis to the neuroinflammatory focus. TSPO is PET against TSPO uses ligands to this TSPO protein that is expressed by the external mitochondrial membrane and is enriched in reactive microglia and macrophages. Another ligand of interest is deprenyl that binds to monoamino oxidase B, that is an enzyme that is enriched in reactive astrocytes. And PET imaging of this deprenyl was able to highlight the epileptogenic area that correspond to area of hypometabolism with fluorodesoxyglucose. Another approach is the quantitative measure of metabolites in the brain that can be done with MR spectroscopy. And for example, myoinositol is a metabolite in astrocyte that can be measured, quantified, and is a marker of reactive astrogliosis. One can measure inflammatory blood or CSF biomarkers, some of which, as indicated here, have had the first validation in the clinical setting as potential diagnostic or prognostic biomarker of predictive of drug responses. And also isolating peripheral blood mononuclear cells, in particular monocytes, macrophages, and to measure their activity to impose inflammatory stimuli. This has been done for Dravet syndrome and for patients with NORSE, and these monocytes in cultures, when exposed to an pro-inflammatory challenge, express higher level of inflammatory molecules that corresponding cells from healthy patients, and a decrease of anti-inflammatory mediators, which again sustain the evidence of reduced resolution response in epilepsy. Very briefly, if I still have 5 minutes, about the mechanism. So a question is, but how these inflammatory molecules contribute to hyperexcitability phenomena and to seizures? Several preclinical studies have highlighted that inflammatory mediators can lower seizure threshold because they alter the homeostasis of synaptic and intrinsic neuronal excitability by activating their receptors expressed by neurons, glial cells, and brain endothelium. For neuronal cells, inflammatory molecules can act very rapidly through non-transcriptional mechanisms, so post-translational modifications, by altering voltage-gated and receptor-coupled ion channels and contributing to the imbalance between excitatory and inhibitory neurotransmission. In astrocytes and microglia, these inflammatory molecules, as I already mentioned, can alter their homeostatic properties, and in endothelial cells they contribute to blood-brain barrier damage. That is another hallmark of drug-resistant epilepsy tissue that can contribute to neuroinflammation and alteration in the excitability of neuronal networks. Indeed, just to mention 2 relatively recent papers where IL-1 beta has been shown to decrease the GABA-A-mediated currents in tissue of patients with drug-resistant epilepsies, while IL-10, that is a pivotal anti-inflammatory cytokine, enhances these currents, but its effect is precluded by IL-1 beta. Again, underscoring the important balance between pro-inflammatory and anti-inflammatory molecules. We have shown, and this is a schematic summary of our activities, research activity in this context, there is a specific interaction between high mobility group box 1 and IL-1 beta and glutamate receptors, particularly extrasynaptic glutamate receptors. So when these cytokines are released by reactive astrocytes and microglia and activate their receptors on neuronal cells, this can lead to a rapid post-transcriptional mechanism which involves the phosphorylation of the NR2B subunit of the NMDA receptor. This is a subunit that is pivotal for calcium entry into neurons, and the activation of this pathway here increases calcium entry into neuronal cells, contributing to excitotoxicity. There are also transcriptional changes in genes that are involved in voltage-gated and ligand-gated ion channels. This has been well demonstrated by inflammatory challenges activating pattern recognition receptor and mimicking bacterial or viral infection, both in adult and immature rodents. And with this one-shot inflammatory challenge, it was found that voltage-gated and ligand-gated ion channels can be modified long-term until adulthood, and this is associated with changes in seizure threshold. Importantly, cytokines can also contribute to structural modifications in brain tissue because they can affect neurogenesis and they can promote excitatory synaptogenesis and reduce perineuronal nets around fast spiking GABA neurons, effects that are mediated by molecules released by reactive astrocytes such as TGF-beta and thrombospondins that are components of the extracellular matrix. And this give rise to increased reactive synaptogenesis, excitatory synaptogenesis, and the reduction of perineuronal nets around GABA neurons can reduce the activity of these inhibitory neurons, contributing to structural alterations in neuronal network that are underlying hyperexcitability phenomena. So I will then stop here, by summarizing with this schematic drawing what is the take-home message. The take-home message is that in acquired injuries, but also in genetic mutations that I have not had the time to go through, there is, together, of course, with the concomitant risk factors, there is the possibility that neuroinflammation is activated. When this neuroinflammatory response is activated and is not properly resolved, then can contribute to epileptogenesis. And again, this neuroinflammatory response persists in animal models during the epileptogenesis phase until the clinical onset of epilepsy. And we still see the neuroinflammatory response in chronic epileptic animals and in chronic epilepsy tissue from drug-resistant forms of epilepsy. The interventions in animal models with some clinical validation supports that this neuroinflammatory response contributes to seizure frequency and severity, drug resistance, and also neurological comorbidities, particularly anxiety-like disorders and cognitive deficits. Again, I didn't have the time to show you the data, but there is increasing evidence in the literature for that. And this means that we can target the neuroinflammatory response at different stages of disease development in the attempt to reduce the pathologic outcomes. We can target molecular pathways with specific intervention, but we can also think of targeting the inflammatory cells that are pivotal for this persistent neuroinflammatory response, such as reactive microglia and astrocytes. Which is the impact on clinical care? If we find effective anti-inflammatory drugs for patients, then we may reduce the probability of seizure recurrence, afford the neuroprotection, improve comorbidities, and have an impact on drug resistance. So I would like to finish here by thanking you very much for your kind attention, for thanking the laboratory, people who contributed to these studies, and my colleagues and collaborators in Italy and abroad. And thank you very much again.
Thank you very much, Annamaria. Super nice talk and super, putting all the things together was super nice. It's open for questions. If anybody has, just speak or raise the hand as you prefer.
Yes, I have a question for you, Annamaria. What about the use of so-called classic anti-inflammatory drugs like ibuprofen or aspirin in patients or in animal models?
Ibuprofen and aspirin have been tried in animal models and they have therapeutic effects. Ibuprofen on acute symptomatic seizures, it was not tested in epileptogenesis models. Aspirin was tried in animal models of epileptogenesis with some therapeutic effects. There is a clinical trial with aspirin in a syndrome that is linked to epilepsy, where they found therapeutic effects, but nothing more than that has been done. Aspirin is targeting cyclooxygenases and is one of the targets that have been developed in the preclinical setting with also specific COX-2 inhibitors and prostaglandin E2 receptor antagonist that might have less side effects that by blocking cyclooxygenase itself. So this prostaglandin E2 receptor antagonist appear to be very effective in model of status epilepticus to prevent the comorbid behavior developing after status and providing neuroprotection. And there are also data about involvement of COX-2, as I mentioned before, in epileptogenesis. Of course, the clinical literature shows that also broad spectrum anti-inflammatory drugs such as steroid or dexamethasone have provided therapeutic effect, particularly in pediatric epilepsies, And dexamethasone, intrathecal dexamethasone is also another approach for inhibiting drug-resistant status epilepticus in NORSE patients, in patients that do not otherwise respond to other treatments.
Okay, thank you very much.
There is go Alberto, go first, and then we have a question from the chat. But go Alberto first.
Hello. First, hello, Annamaria. I saw
Alberto.
you 22 years ago in Seattle.
Yes, nice to see you.
Yeah, nice to see you. Your conference is too amazing, of course, and your experience. You mentioned the cytokines as markers to indicate the potential pharmacoresistance. Particularly, you mentioned interleukin-6 and the relation in the pathway, the interleukin-6 with JAK2 and STATs, the pathway signaling. This signaling increase the synthesis of one inhibitor of ferroportin, you know? For this reason, the iron is accumulated inside the cells, all the cells affected with the signaling of interleukin-6. And it could be related with the ferroptosis, cell death, and consequently, the more inflammation and more seizures. You have any strategies to inhibit the interleukin-6?
There is a recent paper that was published, I think, in Nature Medicine, of tofacitinib, that is a drug,
Exactly.
yes,
Yeah.
that blocks the STAT3 pathway associated with IL-6. They have, tested in their paper that the therapeutic effect of this drug is mainly through inhibition of IL-6 signaling. And but they did not tested ferroptosis or the accumulation of iron in the brain in their model. But I think this could be one drug that is promising because it's a repurposed drug to test your idea. Otherwise, there is another drug that has been published by many years ago by a colleague of mine that was the first showing that IL-6 signaling could be implicated in epileptogenesis. Now I don't remember her name, but it was a drug that had a poor penetration into the brain. So I would not know any other better than this to be tested.
And talking about the monoclonal antibodies treatment, you also mentioned the complement involved in the process, particularly in the refractory status epilepticus or super refractory status epilepticus. Perhaps the addition treatment with eculizumab to inhibit the fraction 5 of the complement could be helped to improve the evolution of these patients, you know?
Yes, I know. I mean, it's one of the potential target to control refractory status. It has not been tested as far as I know, but maybe I miss some literature on that. And of course, it could be tested, of course, yes. But I'm not aware that anyone has done studies so far.
And please, don't forget PGP, please.
No, I don't. Also because, as you know, both toll-like receptor 4 and COX-2, prostaglandin 2 receptors are involved in PGP upregulation.
Some markers you show induce the expression of PGP. Some of your shows, HMGB1, interleukins, COX-2, blah 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 blah. So much.
Yes, no, I did not forget about PGP, but unfortunately all the others forgot about it because after such a, intensive research and try to clinical translation, then it was forgotten about. I don't know why, but maybe there was not a good drug to prevent it from upregulation. I don't know.
Great.
Okay, let's go with the question in the chat. Dante asked, could astrocyte contribute to systemic inflammation in epilepsy? And I am summarizing. He's focusing on molecules coming up from astrocytes outside the brain and activating pathways that lead to comorbidities in epilepsy. And that's the question. The complete question is very long because that's why I summarize that.
We do not know, but it's not something that we should forget about it, because we know that in epilepsy there are, after acute brain injuries, but also in epilepsy, there are astrocytic markers that increase in the blood, in the serum, such as S100 beta that does not come only from astrocytes, also from the liver, but also GFAP and fragments of GFAP that induce autoantibodies in the patients, particularly in traumatic brain injury. So there is a leakage of brain molecules that go into the circulation, but whether they contribute to comorbidities or systemic dysfunctions has not been assessed so far, but it's an interesting idea.
Okay. Any other question?
Yes, me. I have a comment.
Yeah, go.
In 2018, we evaluated the effect of cromolyn, a drug that stabilized the mast cells. When we pretreated animals before the status epilepticus, it provokes a reduction of the histamine release during the seizure activity, and also it reduced the cell damage. When we applied chronically cromolyn subcutaneously using osmotic pumps, we observed that rats that presented the status epilepticus long-term after present less damage and less number of seizures. I think cromolyn could be also a good drug to avoid and to prevent the damage, because mast cells contains cytokinins, histamine, serotonin, and many other substances that induce damage. I want to know your opinion about that.
Well, I did not touch upon the contribution of peripheral immune cells because of a matter of time. Of course, there are circulating blood cells, both immune and non-immune, that are altered in epilepsy. The data are a little bit contrasting because, as you might know, someone has detected modifications in one direction. It was not confirmed because it went into the other direction, which is the role of each of these cell components to the pathology has not been clarified yet unequivocally. But of course, when you have an innate immune response, also the peripheral system is modified. In NORSE, there are many alterations in circulating cytokines as well in peripheral blood. So, I cannot answer your question because I never studied this type of cells, but of course, we have to take into account that also the peripheral cells may contribute. We know that macrophages contribute to neuronal cell loss, neutrophils contribute to neuronal cell loss upon their entry into the brain. It's an interesting observation. I don't think that it has been pursued in the literature very much, which is a pity probably.
Thank you. Thank you.
Any other question? I have two, but I don't want to use my privilege as coordinator of. If no any other question, I have two questions. One is we've been discussing for a long time in the lab the losartan, because losartan seems not to enter to the CNS, but there are proof that it's anti-epileptogenic, at least from that large human study in Germany of hypertensive patients, and we've been discussing why losartan from the periphery is acting. Is really the blood-brain barrier closed during epilepsy or in epileptogenesis, or there is some opening of the blood-brain barrier?
During epileptogenesis, there is an opening of the blood-brain barrier, I mean, increased permeability. And it's most work done by Alon Friedman and on losartan. It seems that losartan antagonizes the effects of the activation of TGF-beta signaling. TGF-beta signaling is activated by serum albumin that gets into the brain upon this increased permeability of the blood-brain barrier. And losartan, according to Alon Friedman data, would block the ability of albumin to activate the TGF-beta signaling in perivascular astrocytes and prevents their inflammatory and dysfunctional phenotype. So this is the underlying hypothesis. It's probably, it does not have facilitated access as many other molecules, but according to his data, the effect would be mediated by this signaling that suggests that it should enter the brain, at least the perivascular space, in order to block this signaling in astrocytes.
And the other question is why these inflammatory mechanisms are not stopped? I mean, because in acute brain injury, you have the dump release, HMG1, everything going out, and then you have a reaction, and then with the time, the reaction is going down because there are several mechanisms of the immune system to reduce that inflammation and not persisting in inflamed way. And why in epilepsy that's not going on? It's because of the seizures, the continuous seizures that it's another release of HMG1 and other dumps, or is because it's a different immune disease, and we are studying like a CNS disease, and it's an immune disease, the epilepsy.
I think that this regulation of the mechanism of resolution is a typical feature of epilepsy that might not be reproduced in other conditions, I don't know. But why, for example, IL-1 receptor antagonist is not induced to a significant extent? It's a question of epigenetic modifications that occurs in epilepsy and they lead to dysregulation in gene transcription, I think. It's one of the main mechanisms. MicroRNA, as I mentioned before, are dysregulated and they may, may boost the inflammatory arms rather than the anti-inflammatory. It's counterintuitive because one should think that the brain should defend itself and not promote what is damaging, but is a dysfunction, that I think also give us some hints about therapeutic intervention, because to prevent the neuroinflammation from occurring is very difficult. It's a very fast event and it's practically impossible to prevent it. But if we can improve the ability of the brain, the resilience of the brain, its homeostatic response by boosting the anti-inflammatory mechanism, then we may be able to stop it short enough and prevent the detrimental consequences.
Any other question, Alberto?
Yes, Javier, there are many questions through YouTube.
Oh, okay, but I cannot see them.
I'm asking for them. For a
Okay.
Alberto can ask.
Go, Alberto, go first. Yeah.
¿Puedo preguntar?
Sí, yes.
Annamaria, you're published with with Eleonora Aronica a couple of years ago, a paper about the action of astroglial in the process of the progress of the epilepsy. And more recently was published another article indicating the activation of microglial, releasing microvesicles that are you focusing to detect these microvesicles with different phenotypes to better follow up and diagnosis the risk of refractory epilepsy and the progress of this pathology or therapeutic responses?
I'm not personally working on this topic. I know that there is an attempt to measure these extracellular vesicles that also contain microRNAs and other biological molecules as biomarkers, but it's not something I am involved with. Although, I mean, it's interesting because you can recognize whether they come from microglia, astrocytes, neurons, because they have markers, so to say. So it could be an interesting approach, but it's not my field of expertise.
I will read some questions by YouTube. Is there any known connection or ongoing research on neuroinflammation induced by COVID-19?
We know that there is this cytokine storm and in the periphery. I'm not aware whether some specific studies on neuroinflammation have been developed post-mortem, or I think that the CSF measurements have been done with indications of increased cytokines. But I wouldn't know, I'm sorry.
Okay.
I have not followed the field very much.
Thank you. There is another question. Are there lymphocytes implicated in neuroinflammation in epilepsy?
They're not implicated in the sense that they cause a neuroinflammation. Neuroinflammation is an innate immune mechanism, is not an adaptive immune mechanism, so it does not depend on lymphocytes. But the lymphocytes can be recruited in neuroinflamed tissues, of course. And there is evidence in focal cortical dysplasia type 2B that there is penetration of CD8, CD4 positive lymphocytes, Rasmussen encephalitis, of course, other malformations of cortical development, tuberous sclerosis, not so much in mesial temporal lobe epilepsy, so it appears to depend on the type of epilepsy. And of course, when you have cytotoxic CD8 positive lymphocytes that attack neurons, they can contribute to neuronal cell degeneration.
Another question. Which of the new anti-epileptic drugs have been developed specifically for the use in the immature brain? I assume it's anti-inflammatory drugs.
Can you repeat the question? Sorry,
yes,
I missed.
I think Anais is asking about anti-inflammatory drugs.
In a pediatric population?
In the immature brain. Have you have been tested in the immature brain?
In humans, you mean?
Yes.
In humans, as far as I know, no new drugs. What is in medical treatment are anti-inflammatory drugs such as steroids, ACTH for infantile spasm, dexamethasone, so more classically anti-inflammatory treatments. But as far as I know, there are medical use, there are no specific new anti-inflammatory treatments, but I might be wrong because I'm not a pediatric neurologist, so.
Okay. Other question. In the chronic phase, controlling seizures seems to be the mechanism for reducing inflammation, or would it be preferable to use an anti-inflammatory drug?
Okay, so it's a question difficult to answer because we know that the recurrence of seizures, even in the absence of tissue structural changes such as neuronal cell loss, so the recurrent seizures per se, non-lesional recurrent seizures, can induce an inflammatory response, okay? But it's not only the recurrence of seizures. In patients with malformations of cortical development, for example, if you compare focal cortical dysplasia type 1 with focal cortical dysplasia type 2B, and we take patients with the same number of seizures, same seizure recurrence. In type 1, there is no measurable neuroinflammation. In type 2, there is, and they are different in structural modifications. Type 1 is cortical dyslamination, and type 2, they have dysmorphic neurons, balloon cells. So it's not only the recurrence of seizures, it's also the structural changes in the brain tissue that matters in order to evoke the neuroinflammatory response. If you reduce seizures, you can reduce the extent of neuroinflammation due to the recurrence of seizures. If we reduce seizures, for example, in our animal models, we see a reduction in the activation of microglia, not so much of astrocytes. Astrocytes appear to be quite independent on seizure recurrence, while the activation, reactivity of microglia is more dependent on the recurrence of seizures. So probably the quote, the extent of neuroinflammation deriving from microglia, if you reduce seizures, will be decreased.
Okay. Other question, in your experience, does inflammation contribute equally when brain damage occurs during development or in adulthood?
I did not understand. Say it again.
In your experience, does neuroinflammation contribute equally when brain damage, such as status epilepticus, occurs during development or in adults?
I see. Okay, the immature brain is susceptible to a neuroinflammatory response. We have tried that with kainate-induced status, depending on the age of the animals. It means that if you induce status epilepticus around 2 weeks, postnatal weeks, you have an inflammatory response. I'm talking about kainate-induced status that is more transient than in adolescent animal, P21, and you don't evoke any inflammatory response in younger animals at P7. This appears to be because kainate in young brain do not activate the transcriptional factors that are pivotal for the neuroinflammatory response. However, if you induce febrile seizures, even in P7 animals, you have a prominent and persistent inflammatory response that contributes to epilepsy, not to cell damage, because it's not observed in this model. So it depends on the trigger of seizures, it depends on the age of the animal.
Okay, and the last question. Given that many repurposed drugs act systemically and may have limited blood-brain barrier permeability, how can we distinguish central from peripheral mechanisms when explaining their anti-seizure efficacy?
Okay, in the animal models, many of the interventions have been also done by intracerebral application of drugs. So we know that IL-1 receptor antagonist, if intracerebrally applied, blocks seizures. And this can be also achieved by systemic administration. and reduces the neuroinflammatory response. For other drugs, they have been given systemically, and we detected a reduction of the neuroinflammatory response in these animals, also before they developed the disease, or before they developed the spontaneous seizures. We do not see any pronounced systemic inflammatory response in the models that we have used so far to test the anti-inflammatory drugs, that are models where status epilepticus is induced by intracerebral application of convulsive drugs or by electrical stimulation. We do not see any systemic inflammatory response in these models. If you use systemic pilocarpine, you have a pronounced systemic inflammatory response because pilocarpine activates T lymphocytes to start with, and they release inflammatory molecules that open the blood-brain barrier and allow enough pilocarpine to enter the brain and cause seizures. So it really depends on the model that you are testing.
Thank you. No more questions by YouTube.
Okay, perfect. I think that was enough, Annamaria. It was we had you like an hour and 20 minutes here, so it's like it's enough.
It's nice. No problem. I enjoyed it.
Okay. If there are no other questions, I think no. Okay. So thank you very much. Thank you very much for coming with us.
Nice to see you, Annamaria.
Thank you, and it was nice to see you. Stay in touch. Thank you very much.
Thank you. Bye bye. Thank you very much.
Bye bye.
Thank you.